Welcome on the hour to the Florence Mahoney Lecture on wisdom on aging from the RNA world, and honoring one of our own intramural staff from NIA. I'll tell you a little bit more about her in a moment. Just a word or two about Florence Mahoney, who don't know of her. She was born in 1899, lived till the age of 103 in 2002. She was a very prominent figure in support for NIH. Her home in Georgetown, actually at the site of some famous movies too, was a soiree location for many politicians and allowed her to exert a great deal of influence. So if you see The Exorcist, the stairs in that are right at her former house. And she was up to the age, of the end of her life, at 103, when we would meet and she would talk about these lectures and about the science, how exciting it was. She wanted to know who was speaking, what it was all about. So a great tradition. Today, it's our pleasure and privilege to have speaking here Myriam Gorospe. She is the head of section and lab of RNA biology and genetics genomics within our intramural program. She did her undergraduate work in Madrid, then came to do her PhD work at SUNY, and a year after that, she came to NIA, and so she has remained to our delight. In addition to her roles in scientific leadership, she's the head of faculty resources and development, and as a leader in many aspects, in addition to her own outstanding and magnificent science. So with that, let me turn it over to you. Myriam, delightful to have you here.
Thank you, Richard, for that generous and kind introduction. I'm really pleased and excited to share part of the research in my lab as in this year's Florence Mahoney Lecture on Aging, and at the same time I'm humbled to have the opportunity to do so. Before I get to the title that maybe some of you think is intriguing, let me just say that our lab's work fits squarely within the mission of the NIA to reduce, delay, and prevent age-associated disease and disability, and really to get away from these age progression schemes that end with someone with severe disability and maybe instead have older age represented by active and healthy individuals. As you see in these two photographs, these happen to be my grandparents. They were both farmers. My grandfather actually was a lifelong lover of rowing. He, in fact, was an Olympian. He was in the Olympic team of Spain. And here he is in his 70s, still going out with the young men, the young rowers in his village in northern Spain. And here's my grandmother with one of many great grandchildren she had. This is actually my oldest son many years ago. So towards the goal of reaching old age healthy and active, my lab contributes expertise, knowledge, approaches, and methods of cell and molecular biologists. And so this lab that I have the great privilege of working with is really the first slide that I want to show you, and in order to acknowledge their contribution and to thank them for the work. The lab comprises many permanent staff as well as many trainees. Down here, sorry. We also work very closely with other subgroups within the section, including the translational senescence unit, led by Ali Herman, who's an independent research scholar, the first, in fact, in the NIA IRP, and with her group, as well as with Supriyo De, who heads the computational biology and genomics core, and also with his group. And so to accommodate this diverse group with a great deal of talent, but also very different interests, we have a program that is correspondingly diverse, and I'm just going to give one slide with an overview of the research in the lab. We have 4 project areas. In one, we look at changes in body composition as we age. We are also interested in changes in our ability to metabolize energy, to sense energy, store, mobilize, etc., as we age. We're also interested in the progressive decline with aging, in our cells' ability to respond to stresses and mitogens, and we're interested in cell and molecular aspects of neurodegeneration. Okay. And these 4 areas are quite different, but they are common in important ways, and one of them is in how we study them, and that is by focusing on gene expression programs that underlie these age-related differences. And so to very briefly review the process of the flow of gene expression, it begins with the transcription of DNA into RNA, but then this RNA undergoes further modification, splicing, maturation, export. It may be stored, transported in the cytosol. It may then be subject to decay, or may, in fact, reach the translation machinery, the ribosomes, in very precise amounts at the right developmental stage, in the right part of the cell, at the right time, in the right amount, et cetera, so that the proteins that are produced match the needs of the cell at the time. These proteins continue this flow of genetic information by undergoing modification, turnover, transport, secretion, and even export out of the cell, as in extracellular vesicles. Much of the focus of the lab is actually on the RNA molecule itself, and really our approaches focus on how the fate of a messenger RNA is dictated by factors in the cell, particularly RNA-binding proteins and a vast and growing class of non-coding RNAs. Some of them are small and typically bind the mRNA directly, and others are longer. They're long non-coding RNAs, so link RNAs. They may be binding directly the RNA, or maybe doing things outside of the messenger RNA, but otherwise influencing mRNA fate. And so this focus on so these RNA protein complexes, these ribonucleoprotein complexes, are really the focal point of all the projects in the lab. And so back to the title. So I thought that in today's talk, I would present, or I would propose to you that by studying, that we can gain information about old age or old human age by looking at these old molecules, these RNA molecules that in the views of some were actually the original holders of genetic information. And so what I'll be telling you is that we can learn about aging by studying these ancient molecules, and in the process learn about young roles or novel roles that we didn't know RNAs did before. All right, so according to the RNA world hypothesis, after Earth formed some 4.5 million years ago, and a stable hydrosphere was established, the first complex molecules began to form, and some of them began to aggregate in these pre-RNA-like molecules that were likely short and likely labile. But over time, probably through chemical modification of the RNA and association of the RNA with molecules, with other molecules, these RNAs became more stable and, in fact, became capable of replicating themselves as well as having a range of catalytic activities. Okay? So here we're looking 3.8 million years ago. But soon thereafter, relatively speaking, the DNA molecule evolved. This is now double-stranded as compared to the single-stranded RNA. And this double-stranded molecule was more stable, was a better container for genetic information, and was also, importantly, more faithfully repaired if that DNA became damaged. And from there, we had viruses and unicellular organisms develop and life, really, with the diversity of species that we know today. So, but RNAs have remained these vital regulators of life and gene expression today. And so when we think of RNAs, I think most of us think of coding RNAs, messenger RNAs. These RNAs are the ones that are studied the most extensively. They, of course, encode protein. But in fact, the vast majority of RNA synthesized from our genome is non-coding. Right, and this very diverse class, this is one classification, there are others as well. The ratio of one versus the other is as follows. Messenger RNAs are only expressed from 2% of the genome, whereas 98% of the genome actually produces or transcribes non-coding RNA. And so today I will be talking about some of these regulatory non-coding RNA, these link RNAs that I mentioned earlier, as well as microRNAs, and how they influence aging by focus on a specific part of the program. So we'll be looking at RNPs and really focused on only this project area. And in fact, the process of cell senescence has been the topic of other Florence Mahoney lectures. So it is a central theme in aging biology today. So what I'd like to do is walk you through three things. One is introduce cell senescence a bit more formally, or, well, introduce cell senescence, I haven't done that yet, and then offer 2 examples of how we learn about aging pathologies by focusing on senescence and senescence-associated gene expression, and then end with a few very brief vignette-type examples of how regulatory RNAs are giving us insights into senescence. So cell senescence is one of those hallmark traits of aging. They are represented in this schematic that is taken from this Hallmarks of Aging review that just came out by Lopez-Otin and colleagues. It follows up on an earlier landmark review by the same authors, again, looking at different traits of old cells in tissues and organs. And you can see, probably recognize many of these other changes that also occur with age. So cell senescence is a process that we and many other labs have tried to catalog and really quantify, and in order to study it and see how it changes and over time in tissues. And this is one such attempt, just to illustrate this point. So here are 3 tissues from young donors. The study had about 12 or so tissues that we looked at. And in a young person, we don't see a ton of senescent cells. They will show up brown, as you see here, because we are detecting P16, and that's what this signal is. But in response to a range of damages, typically sublethal damage that occurs over the lifetime, we see the emergence of senescent cells in older tissues. So these are the same organs, but now using tissues from older donors. And now you see many more senescent cells across these tissues. Examples of these sublethal damages include developmental signals, genotoxins, oxidative stress, telomere dysfunction, oncogene activation, etc. Now, these cells that accumulate now in older tissues are non-dividing. They have unresolved DNA damage. They express P16, P21, P53, a number of growth suppressors. They have a very active lysosomal compartment with a senescence-associated beta-gal activity that is one way of detecting these cells. And they have a robust senescence-associated secretory phenotype, or SASP, which is critical because it includes production of factors such as cytokines, growth factors, matrix metalloproteases, and other proteins that, even if expressed by a handful of cells and in even low concentrations, they are such potent bioactive molecules that they really change the function of the organ and sometimes also the architecture. Now, we can study senescence in tissues, but for some questions that we might want to ask, it's easier to work with culture cells. And we can trigger senescence in culture by taking primary cells in general. We can also do this with cancer cells and other cells, and exposing them to the same types of triggers that you saw earlier, and they become senescent. Here we're detecting senescence by this beta-gal activity that is lighting up in blue. And in this case, Gabriel Casella, who led this work, a postback in the lab, was doing replicative senescence, exposure to oxorubicin, exposure to ionizing radiation. And these cells have the same features that I showed in tissues. They're non-dividing. They have unresolved DNA damage. They express P21, P53, P16. They're beta-gal positive, as you see here in blue. And they display the SASP. Now, senescence as a biological process has been selected over evolution because it plays really key roles in physiology. They're important for developmental morphogenesis during embryonic stages. After birth, they're important. Senescence is important for wound repair, for tumor suppression, particularly in young individuals. But over time, as the exposure to these sublethal damages continues during the lifetime, senescent cells accumulate. And so in old age, senescent cells have a very adverse effect, a negative impact. And in fact, the accumulation of senescent cells during aging has been linked to the development of many age-associated diseases and declines, such as sarcopenia, arthritis, cardiovascular disease, obesity, type 2 diabetes, cataracts, COPD, tumor promotion now in old age, neurodegeneration, immune dysfunction, and others. So there has been really a growing recognition that probably eliminating these senescent cells might be beneficial in aging in aging phenotypes. And in fact, this idea was solidified and really supported by a landmark paper by Darren Baker from Jan van Duurster's lab at the Mayo Clinic, who provided evidence for this. So these two mice are littermates. They're 9 months old. In these mice, senescent cells are allowed to accumulate. These mice have a premature aging. They're genetically modified to have premature aging. But as you can see in this littermate, eliminating senescent cells in the presence of a drug that is abbreviated here with AP leads to a very healthy phenotype in old age. So these mice now compare with the old counterparts, which show kyphosis and graying of the hair and loss of vision and fat and muscle. The mice in which senescent cells are eliminated preserve muscle mass and function, adipose tissue, vision, etc. So this and other models that have been reported since then have led the field to conclude that selectively eliminating senescent cells promotes healthy aging. And so moving then to pharmacology, we now have a number of agents known collectively as senolytics that accomplish the same effect, that can eliminate senescent cells selectively and leave untouched those cells that are non-senescent. And here too, the list is just a little bit longer than this. So we don't have a ton of senolytics today, but we do have a good number, including these two, dasatinib and quercetin. This is a flavonoid. This is a cancer drug. They're typically given as a cocktail, and when given together, they again eliminate selectively senescent cells, and the end effect is, as we saw in the genetic model, to promote healthy aging. And I mentioned these two also because I will get back to them twice in my talk. And so let me tell you a little bit about aging pathologies that we have in which understanding senescence and senescent gene expression programs has been informative and quite valuable. So I'll present two brief examples, one looking at neurodegeneration, the other one at autoimmunity, both of them increasing, of course, with old age. So Alzheimer's disease is a devastating neurodegenerative disease in which there's massive neuronal cell loss, and this loss is linked, at least in part, to the presence of toxic aggregates, such as those depicted here, amyloid beta plaques. And so in this study, we asked a very specific and kind of focused question. We asked, in this adverse environment of the AD brain, do we see the sublethal toxicity that might give rise to senescence in particular of cells that support neurons, right? And so we focused on astrocytes, microglia, and oligodendrocytes. So this work was led by Pei-Su Zhang in Mark Matson's lab when he was still at the NIA IRP. And what Pei-Su found is that when looking at brains from a patient who had died with and had Alzheimer's disease, that embedded in these A-beta plaques were these cells that stained positive both for a senescent marker, P21, and for an oligodendrocyte marker, OLIG2. Okay, so this led us to conclude that that OPC, oligodendrocyte precursor cells, are the major senescent cell population in human AD plaques. And so to study this population in a bit more detail, Pei-Su moved to a mouse model in which we could ask a few more questions. And so this is a mouse model in which plaques are developed through transgenic expression of a mutant form of human amyloid precursor protein, APP, as well as presenilin-1. An age-matched controlled mouse does not show any AD-like plaques in the brain, but in this transgene we do see that many plaques appearing by 7.5 months. An enlargement of that field shows that we see these plaques, and also we see presence of beta-gal activity. That's this blue hue that we detect. So we decided to study in a bit more depth the cell content of these foci. And so we used laser capture microdissection to cut out the region surrounding the plaque, isolated RNA from these discs. And then by reverse transcription and quantitative PCR analysis, we look for markers in these tissue, in tiny tissue discs. And so we found markers of AD, markers of oligodendrocyte precursor cells, and markers of senescence. Again, supporting this idea that we had senescent cells in this niche as well as OPCs. So then Pesio was ready to ask the question that we really wanted to ask in mice, which was, if we eliminate senescent OPCs, do we then see any changes in AD pathology? And so she treated these mice once weekly for 11 weeks via gavage with D+Q and with senolytics. And at the end of the 11 weeks, she did a number of tests. I'm only going to show 3 of them here. So first of all, she found that compared with vehicle-treated mice that received the senolytics had fewer plaques, and these plaques were smaller and showed low evidence of having senescent cells. When she tested plasma, she found reduced levels of factors that are typical components of the SASP, again, in the senolytic-treated mice. And very importantly, when she looked at learning and memory, which, as you know, is the loss of which, as you know, is a hallmark of AD, she found that mice that received the senolytic again performed better. So the vehicle-treated mice swam around this pool in this Moritz water maze, not remembering where the platform had been placed, whereas mice that received the senolytics had a preferential swimming in the quadrant where the platform existed previously. And so improved learning and memory. So this was really quite exciting and led us to conclude that senescent OPCs are found in AD plaques. Senescent cells can be eliminated by senolytics in the AD niche. Treatment with senolytics improve key AD-associated traits in this mouse model, and use of senolytics warrants consideration in AD therapy, we propose. And I just, as I was preparing this presentation, I found this study that seeks to test the effect of senolytics in cognition and mobility in older adults with at risk of Alzheimer's disease, and the results will be reported or will be finished this year, so hopefully we will hear soon. All right, moving to the second example in which understanding senescence has proven to be quite insightful and important. I'll switch to autoimmunity. And for this study, I will offer for background the Sardinia study. This is a study that has been ongoing for 22 years that aims to identify longevity genes and the genetic basis of age-related traits and diseases. And the study is based on the island of Sardinia, off the coast of mainland Italy. This is an island that has been relatively protected from invasions, and so the gene pool of Sardinians is relatively pure, and it makes it easier to identify disease-causing genes, including genes influencing aging traits. And so there has been a long-standing collaboration between the laboratory, the scientists in the Sardinia team and the NIA in Baltimore. And one of the studies resulting from this collaboration was published here in 2007. And here we found that the cytokine BAF was linked to autoimmunity on the island. Okay. So BAF is one of those hallmark traits of the SASP. Whether we look in tissues or in tissue culture, we always find BAF as one of the key components of the SASP. In fact, Martina Rossi, a current postdoc in the lab, just dedicated an article to characterizing BAF as a marker of senescence in the SASP. And so in this study, the interesting discovery is that there was a variant of BAF that was linked to multiple sclerosis, which is quite prevalent on the island, right? And the interesting first discovery linked to this GWAS analysis is that persons who had both variant alleles had high levels of BAF. And this was in contrast with persons who had both wild-type alleles and had much lower levels of BAF. A heterozygote person had intermediate levels. And the only difference between these three genotypes is the length of the 3' UTR that encodes the BAF. So here is the messenger RNA that encodes BAF. We have a 5' untranslated region, or UTR, and a coding region, and a 3' UTR. And the only difference between the wild type and the variant is the presence of an early polyadenylation site, an insertion, that with high frequency makes this RNA shorter. All right? And so this was quite intriguing, because actually the protein is identical, expressed from the coding region, identical from one mRNA and the other mRNA. The major difference linked to the length of the 3' UTR is the fact that the longer 3' UTR accommodated 2 sites of repression by microRNA miR15, whereas the shorter version accommodated a single site. Okay, and this alone contributed to these changes in BAF levels. Additionally, we discovered, as these studies progressed, that there was a protein that actually facilitated this repression by miR15. This protein was studied by Laura Ida, who had also led the original study in the New England Journal of Medicine. And what she found, without going into the molecular specifics of the study, she made a synthetic 3' UTR from the long messenger RNA and asked what RNAs bind to this protein. And by mass spec analysis, she found that NF90 was, in fact, the top interacting RNA binding protein with the long 3' UTR of BAF. She then mapped a little bit closer using other molecular methods, some short RNAs, and then looking for binding. And she discovered that NF90 binds at a position on the 3' UTR that is immediately distal to the alternative polyadenylation site. So as you can see, the BAF variant does not bind NF90, but the wild type does. And so putting together all this information, NF90 actually facilitates, in work that is reported in her article, facilitates miR15 mediated repression and low BAF levels, whereas the variant does not have the benefit of NF90 mediated recruitment of a microRNA, and BAF levels remain high. Okay, all right, so having, I hope, made a case for, explain what senescence is and how we study senescence as part of aging pathologies. I'd like to tell you a little bit of how we study senescence by focusing on regulatory RNAs. Okay, so I'll briefly touch on long non-coding RNAs, circular RNAs, and microRNAs. So these ribonucleoprotein complexes have been the bread and butter in the lab, and much of what we've done with them is study whether they promote or suppress the process of senescence. And even though I talk about regulatory RNAs, as you'll see here, many of these are proteins that have critical roles in mRNA fate. And so I do need to at least mention that RNA binding proteins are key regulators of senescence. I already talked about NF90, but there's another RNA binding protein that we've studied quite extensively in the lab, HUR. This is a protein that typically promotes proliferation and promotes expression of many proliferation-associated genes. It's very highly expressed in cancers. And it's almost gone in senescence. We see very, very low levels of HUR. And in fact, simply silencing HUR can trigger senescence on its own. All right, so, but most of what I will tell you in the remaining slides focuses on non-coding RNAs. And so we recently had the honor, but also the challenge of reviewing the past 25 years of our understanding in long non-coding RNA biology. So I'm just going to use this schematic to tell you that link RNAs do a lot of things, a lot of different things. They're exciting, they're unexpected, they're surprising, and for an RNA biologist, they're a lot of fun to study. And so link RNAs can reside in the nucleus or in the cytosol. In the nucleus, most of what link RNAs do have to do with chromosome organization and transcription. So I'll just read to you the categories that we design or that we ascribed. So chromosome organization, chromatin organization, regulation of transcription, splicing, assembly of nuclear bodies, and nuclear export of select molecules. In all these vignettes, the red, of course, is the long non-coding RNAs. Again, they can be linear or circular. In the cytosol, link RNAs do even more diverse and kind of exciting functions. They can modulate mRNA turnover. They can help assemble cytosolic aggregates like processing bodies, stress granules, as well as the cytoskeleton. They can modulate the translation, and they can affect the stability of certain proteins. They can affect mitochondrial function, as well as the function of the endoplasmic reticulum and the Golgi. So really very, very diverse functions, and as I said, often surprising and unexpected. And if you'll indulge me, I'm going to add one thing. And this is something that emerged as we were working on this review, which is it appeared as though long non-coding RNAs provided an additional layer of gene regulation. And I'll just explain briefly what I mean. So at the genomic level, we have DNA that is used for transcription of RNA, and RNA that is used to of course, synthesize protein. And on top of this level, we have epigenomics, right? We have regulation by modification of the DNA, the RNA, the proteins that assemble DNA or bind RNAs. But built on these two layers, we proposed are long non-coding RNAs, which even further refine how these complexes maybe fit with one another or change location in the cell, et cetera. So we call this layer of regulation supragenomics. Okay. All right, I'll just leave that at that. And so I'll tell you about a few of the non-coding RNAs we've looked at, and I'll end the talk with those examples. So long non-coding RNAs have been studied in our lab for many years in the context of senescence, and we know many changes, increasing or decreasing as cells reach senescence. And I'll say a couple of things about these two. So Hotair is a long non-coding RNA that we studied about 10 years ago, and this link RNA increases in senescent cells. It's almost undetectable in proliferating cells. We find it in the nucleus, but we also find it in the cytosol. These dots, these fluorescent dots. Here we're using RNA fluorescence in situ hybridization, RNA fish, to see them in collaboration with John Rinn and Arturo Orjalo. And as we look deeper into this RNA, Jae-Hyun Yoon, a former postdoc in the lab, discovered that this RNA binds many proteins, including HUR. And binding of HUR actually leads to the recruitment of an RNA-induced silencing complex, the RISC, guided by this microRNA, LED7. And the arrival of this complex causes degradation of Hotair. All right, and this is important because when Hotair is stable, it has a very peculiar function, and that is to serve as a, what I write here, as a scaffold for binding of many proteins. And some of them, in fact, these two pairs of proteins are biologically or functionally related. DCP3 and MEX3B are E3 ubiquitin ligases. And these other proteins, ataxin-1 and snurportin-1, all of which have affinity for Hotair, find each other or have become in close proximity by binding the same substrate link RNA, and so ubiquitination is more efficient, and degradation of snurportin-1 and ataxin-1 ensues. So in senescent cells where we have high levels of Hotair, when we look at ataxin-1 and snurportin-1, we see much lower levels of these two proteins, and we also, of course, see more ubiquitination and other molecular details. In cartoon form, proliferating cells constantly degrade Hotair. So these two proteins are actually stable, snurportin-1 and ataxin-1. But in senescent cells, where Hotair levels are elevated, these proteins are rapidly degraded. Okay. Another link RNA we've studied in the context of senescence is link RNA P21. Here, we have a link RNA that also regulates protein expression levels, but instead of changing the half-life of that protein, instead this link RNA controls the translation. And so the study, also led by Jae-hyun, looked at binding of HUR to this lincRNA, which also recruited LET7-driven risk. So this is a shared feature of the two lincRNAs, leading to degradation of lincRNA P21. And when lincRNA P21 is stable in cells, then we see, as we see in senescence, we see that it forms partial hybrids with a subset of messenger RNAs in the cytosol, including CTNNB1, which encodes beta-catenin, and JUNB mRNA, which encodes the transcription factor JUNB. And when these partial RNA hybrids form, there is translational repression and reduced production of JUNB and beta-catenin. Right, so in proliferating cells where lincRNA is constantly degraded, we have active translation of these two proteins, whereas in senescence, where lincRNA P21 leads to the repression of translation of these two proteins, we see far less expression of them. Okay, so this is all I'll say about lincRNAs, and I'll mention circular RNAs again with vignettes, just very simple examples of how we study them. So I'll talk about these two circular RNAs. Kurt Adelbosen, a staff scientist in the lab, very talented, has been in my group for many years, looked at SIRPAVN1 as one of those circular RNAs that emerged from this precursor messenger RNA. So typically, this is the first transcript that arises from transcription, and then, of course, undergoes splicing and produces a mature messenger RNA, in this case, encoding PAVN1, right? So here's the PAVN1 mRNA. But for reasons that we don't know for this circular RNA or for any other, in fact, pretty much in circular RNA, in some cases, individual exons or individual introns, or sometimes combinations of introns and exons or portions of them, recircularize. So this end covalently recloses with this end. So we have a very stable, covalently closed RNA molecule. And so when Kurt looked at SIRPAVN1 in detail, he discovered that actually the entire exon, which was 152 nucleotides long, had many sites. Each of these bars that are all so tightly together are sites where actually HUR bind. So back to this major regulator of senescence. And so SIRPAVN1, we envisioned it as a sponge or a very avid binder of HUR. And this protein actually dimerizes and multimerizes, so it really could be a point where a lot of HUR protein is binding and not available for other functions, right? That's all I'll say about SIRPAVN1, and I'll just say even less about this other circular RNA. But circular RNAs can bind many other molecules. And in fact, the first molecules that were shown to bind circular RNAs were MicroRNAs. And Amaresh Panda, also a former postdoc in the lab, now a PI in India, discovered that SIRPVT1 was actually lower in senescence. So here's one example of a lincRNA that shows reduced levels in senescence. And this was important because SIRPVT1 binds LET7 with high affinity and a couple of sites per circle. But in senescent cells, LET7 cannot be bound to the circular RNA because there's much less of it. And so it's free to bind and repress other genes, including many that encode proliferative proteins. And the last example that I'll share with you today looks at the role of microRNAs in senescence. And I'm going to talk specifically about one of them. And so the work fits within this larger goal in the lab of identifying universal markers of senescence. This is something that many labs interested in looking at senescence have been after. And so this very talented postdoc in the lab, Gabriel Casella, he's now an MD PhD student at University of Chicago. So Gabriel said, well, if I take enough pairs of proliferating and senescent cells from different tissue origins and I render them senescent using different methods, maybe I can end up with an RNA signature of senescence, right? So that was the goal of the study. He compared not just these 4 pairs, or even these 8 pairs. I think he compared 13 pairs And some of them were fibroblasts, others were endothelial cells, epithelial cells. The triggers were ranged from DNA damage, replicative exhaustion, oncogene-induced senescence, etc. And so he sequenced these pairs of proliferating senescent cells and then identified 50 RNAs that were across all the pairs were elevated, and 18 RNAs across all the pairs that were always reduced in senescence. Okay, this report was more of a resource study, but it was also the basis for a study that Ali Herman, who was still a postdoc in my lab, she now, as I mentioned earlier, she's now an independent research scholar. But Ali Herman took these RNAs and said, well, maybe some of these coordinated regulation happens post-transcriptionally. And as we know, microRNAs and other post-transcriptional regulators have the ability of jointly controlling a number of messenger RNAs. And so she postulated that maybe there were microRNAs that might be jointly controlling some of these increased RNAs or decreased RNAs. And so she did a bit of bioinformatic analysis with help from our bioinformaticians in the core, and found these 6 main microRNAs that might regulate some of the groups of RNAs that we knew were increasing or decreasing with age, with senescence. But unfortunately, only one of them actually changed during senescence, and that is MIR340. All right, so she looked at MIR340 in more detail. If she simply overexpressed MIR340, cells reach senescence faster. The starting point here were pre-senescent cells, so these are cells that had endured some damage, but they were still proliferating. And if she antagonized MIR340, what she saw was the opposite, a slowing down of senescence, and in fact, more proliferation. Right, it's here in orange. So she said, well, maybe MIR340 is controlling a subset of these senescence-relevant messenger RNAs. So she sequenced these cultures and then looked at the programs, the changes in messenger RNAs. And many of these changes, many of these changing messenger RNAs encoded proteins important for cell division, DNA replication, chromosome condensation, chromosome maintenance, et cetera. These are things that we think about when we look at senescent cells. So these look promising. So she said, well, how are the messenger RNAs regulated by MIR340 fitting into this larger question of senescence? So she looked first at all the target mRNAs that were predicted bioinformatically to be MIR30 targets. Then she looked at all the messenger RNAs that in her cells were being downregulated during senescence by the treatment with by the transfection of MIR340. And finally, she merged these data with the RNAs that we knew were going down in the senescence transcriptome that Gabriel had identified previously. And so the long regulated RNA encodes this protein, LBR. The LBR messenger RNA has a beautiful binding site for MIR340. Here is the complementarity of the microRNA and the messenger RNA. LBR is a protein that we had never studied before in our lab, but it's part of the inner nuclear membrane and sits in a so-called lamina-associated domain, or LAD, and the chromatin around this region, this nuclear region, is typically silent. It's not transcribed. And this is important. This silencing of transcription is important in order to preserve proliferation and to not precipitate senescence. And so overexpressing MIR340 completely obliterated any LBR signal that we saw in the control group. As you can see, these signals match well the nuclear signal, as they should, because LBR is a nuclear protein. Here is LBR by Western blot analysis. Again, very nice reduction by the microRNA. And simply silencing LBR actually increased trigger senescence very efficiently. And conversely, when LBR was overexpressed, we saw a reduction in senescence markers. So now we see lower levels of P21, P16, and other markers. And so, and I just have one more data slide. From this model, we proposed that maybe Mir340 was triggering senescence, at least in part, by eliminating this pivotal protein in the organization of the LADs, such that then the LAD regions became decondensed and senescence ensued. Okay, so then Ali went one step further and asked, well, maybe can we exploit this ability of Mir340 to really push cells into senescence? Can we exploit this ability in a way that maybe could be therapeutically useful in the future? And so with the data that she gathered, and again, remember, senolytics are effective in cells that are fully senescent. And so she used quercetin and dasatinib, as well as this other senolytic, ABT737, to ask the following question. Can we potentiate the activity, the senolytic activity of D plus Q, as well as ABT737, by further pushing cells into senescence through the action of Mir340? And sure enough, that's what she found. When she looked at cell viability, it was reduced when the microRNA kind of, again, pushed cells into deeper senescence, and now the senolytics were more effective. She looked at the same question of cell viability by now measuring caspase activity, right? As cells undergo apoptosis, caspase activity rises, and again, with the two senolytics, the effect was very poor. But if she then overexpressed Mir340, she saw much more potent senolytic effects of the senolytics. All right, so in summary, what we propose is that Mir340 can push pre-senescent cells into full senescence and then enable senolytics to be fully effective. One might, in some cases, want the opposite, want to see cell survival, and so perhaps antagonizing Mir340 could have that effect on promoting cell survival. All right, so what I've told you from this final third part of my talk is that senescent cells is regulated by many regulatory RNAs. And in particular, I mentioned a few long linear noncoding RNAs, as well as some circular noncoding RNAs, microRNAs, and of course, RNA binding proteins that enable a lot of what the noncoding RNAs, noncoding regulatory RNAs do. And some examples and brief examples of them. I invite anyone interested in seeing more details to look. This is all published work. Okay, so what I've told you today, now wrapping up, is that cell senescence exacerbates many age-associated declines and diseases, that reducing or eliminating senescent cells has a beneficial impact in a model of neurodegeneration, AD in mice, that senescence-associated protein BAF is regulated by the joint action of a RNA binding protein working together with a microRNA. And finally, I presented a few examples of how regulatory non-coding RNAs modulate gene expression programs in senescence. Now taking a step back and kind of going full circle and back of what we know about these ancient RNA molecules, I hope I have presented interesting evidence that RNAs are in fact critical ways of regulating aging-associated processes such as cell senescence. But if I may, I think it also has revealed to us that by studying aging, we are learning a lot of cool things, a lot of novel roles of RNAs themselves. So it's a kind of a two-way research benefit or exciting in both ways, I would say. All right, so I've already acknowledged people in the lab. I also have to acknowledge some of our collaborators with whom who participated in the studies I presented today, Mark Matson and Pei Su Zhang, long-standing colleagues, Luigi Ferrucci, Rafa De Cabo, Nate Basisti, and others. Of course, the Sardinia team, led by Francesco Cucca, and always with the presence of David Schlesinger, who bridges both teams, and former lab members, particularly Weingong Wang, who did the original work on HUR in senescence, Ji-Hyun Yoon, who did some of the lncRNAs in senescence work, and Laura Ida, who did some of the BAF studies. I'll thank everyone, and I will thank all of you as well for your attention, and take questions if there are any. I'll leave this on.
Do have a couple questions online from Joe Ziegelbauer. There you are. Okay, behind the sign. Circular RNA biogenesis can be promoted by NF90. You showed an association of NF90 with BAFF RNA. Do you think NF90 influences circular RNA biogenesis from the BAFF transcripts?
Thanks, Joe, for that question. We have not tested that. I don't know, but it's certainly something we can look at. And I hope Martina is listening, and we have work to do. I think it's a great idea. Thank you.
Okay.
Hi, Myriam. How are you?
Hi, Mark.
So I think you made a really convincing argument that the circular RNAs change during, and they're both, well, they're certainly sufficient to induce. So I don't know if you've shown that they're necessary. But then how does the cell know that it's aging in order to regulate those microRNAs? And, you know, these various RNA gene expression modules, what's driving that? Do you know?
Well, a lot of the DNA damage response pathways turn on expression of this is a very general response, but turn on some of the long non-coding RNAs that may be important for maybe repairing our DNA, and in the process affect cell cycle regulatory proteins and et cetera. So it would be, I think, just a readout of the energy state of the cell as well as the integrity of the DNA state of the cell. Thank you. Thanks, Mark.
That was amazing. You've shown that there are so many different levels at which RNA is regulating senescence in the aging concept. What I'm trying to understand is, do you think this is co-opting the normal functions of those in different wound healing or in development, or is this something that's unique to in the aging setting? I'm trying to get the balance between those two factors. Do you have any thoughts on that?
Yeah. Thanks for that question, Natasha. It's a great point, and honestly, we don't know. But I think you're right that the cell is sensing something that looks like a wound, but a kind of a wound that extends over time and over a vast amount of space in the tissue or the organ, and is trying to respond to that by recruiting immune cells, by remodeling the extracellular matrix, by doing all the things that you would do to repair damage.
And do you see that there might be a sense of a hierarchy at which different RNA molecules are acting in the context of the senescence? You know, is it that when you're getting to circular RNAs, is that the backup of the backup of the backup? Or is it, you know, is there a hierarchy, or is it just everything's being thrown at it all at the same time?
Hierarchy. I do think there is a hierarchy. Something I didn't mention, and in fact has not been studied much, is that during senescence, the translation machinery is down. And a lot of the transcription machinery is also much lower. And so it's very helpful to have these already made, these RNAs that can jump in, they're already there, and can fill in for the functions that we would otherwise need new transcription to fulfill. So I do think there is a hierarchy. We don't really know what that hierarchy is in detail.
Great. Thanks very much.
Yeah, thank you.
Fascinating talk, Myriam.
Thanks, Max.
So in the old RNA world, before DNA and protein emerged, right, RNA is used to perform all the kind of enzymatic, activities, right? Even in the new world now, some RNA still has that enzyme activity, like self-splicing intron in tetrahymena, right? So in your opinion, do you think, now there is still all the kind of RNAs, microRNAs, non-coding RNA, circular RNA. In your opinion, do you think possible that, some of the RNAs still possess that enzymatic activity?
Yes, no, not just my opinion. Ribozymes are studied actively here in NIH. And so, yes, many have retained that catalytic activity that independent of protein. Yes, definitely. Yes, thank you. Thanks for that question.
If I could just take a follow-up to that related question. I hadn't thought so much about the RNA DNA evolutionary spectrum that you posed, is there evidence or the data to speculate upon whether the non-coding RNAs that are now a part of DNA-driven biology were descendants of original RNA, or was that a dead end? And the RNAs current all a product of the DNA era?
Yeah, I was afraid I was going to get questions about the RNA world hypothesis, because I really don't know, Richard. I'm sorry. I don't know if they're vestiges or they're evolved later, or they. Sorry. Yes.
Hi there. First of all, great talk. I'm fascinated with these circular RNAs that you were talking about. Has there been a lot of probing, either by your lab or other labs, into the broader biology of these RNAs? Like, are they a necessary cofactor for protein polymerization? Do they act to help the proteins move about? What's kind of like the broader biological role of these proteins? I was wondering if you could speak to that a little bit.
Yeah, great. Thank you for that question. I didn't have I sometimes have a schematic of all the things that circular RNAs bind to, and it is a lot of things. So circular RNAs can bind RNA binding proteins, splicing factors, microRNAs, messenger RNAs, long non-coding RNAs, DNA, as well as ribosomes. In fact, some circular RNAs are partly translated. So really, I think to answer the question in a simple way, the function of the circular RNA is dictated by the things that it binds to, by the molecules it binds to, but those molecules are very diverse and complex. So the functions of circular RNAs are really, really quite fascinating and yeah, and a lot more to discover.
Got it. Thank you.
Thanks for the question.
Hi, Myriam.
Hi.
I had a question on your MIR340.
Yes.
So you showed, if I remember correctly, you showed that MIR340 levels are higher in senescent cells,
Yes.
and if you overexpress it,
Right.
the cells become senescent, and if you knock it down, you see the opposite. So I was wondering if you have any clues on how MIR340 expression is regulated during senescence.
Yeah, we don't know.
Maybe
Great question,
it
right?
may be a part of a bigger, network of genes that is important
We
for senescence.
never looked at the host RNA or the transcription factors that might drive MIR340 production. We did not. That's also something we could and should look at.
And along those lines, MIR340 is conserved between human and mouse, is it?
Yes.
So is there a MIR340 knockout mouse, and what's the phenotype?
You know, I don't know. I don't know if there is such a mouse. Sorry. We will look into it. Thank you.
Thank you.
Thanks.
There's another question online that coincidentally combines these last two questions from Howard Young. Did MIR340 bind to endogenous retroviral RNA? Excuse me. Did it bind to endogenous retroviral RNA?
So we did not look at that. But in the process of studying MIR340, and I didn't include that data because I think it would be too complex to discuss quickly. But when we overexpress MIR340 and reduce LBR, we see a lot of repetitive elements, non-coding RNA repetitive elements that are located in the latch, or the chromatin that sits in those lamina-associated domains. Those become expressed. Mm-hmm. And it's not exactly answering the question, but it's related to vestigial RNA that is expressed in response to this microRNA.
Hi, Myriam.
Hi, Chris.
What a great talk. Amazing breadth of presentation. I learned a ton.
A little closer to the mic.
One of the questions I have for you is kind of a big picture question for CNS neurons, so post-mitotic neurons, and the idea of maybe they're just sort of in a rut, and because they're post-mitotic, you don't want to lose them. Is there a way potentially in the future to try and convert them back once the original negative stimulus is subsided, to turn them back into a functional neuron?
Yeah.
So.
Thank you for that question. I think a smarter person than me needs to answer that question. If these neurons can be reprogrammed to sort of restore the function, right? Honestly, I don't know. Maybe someone in the audience can help, but I'm sure there's a lot of work being done in that direction. So, any other questions?
Dr. Hotair, you can wrap up if you like.
Well done. Precisely on the second, I think we've done it. Thank you for a wonderful lecture, which is what we expected from you, Myriam, and for great participation for those here in the room and for those remote. Thank you all. Look forward to seeing you some more Walls lectures. Myriam, thank you.
Thank you.